This Week in Microbiology is brought to you by the American Society for Microbiology at microbeworld.org/TWiM. This Week in Microbiology, episode number 63, recorded on August 29th, 2013. Hi, everybody. I'm Vincent Racaniello, and you are listening to TWiM, the podcast that explores unseen life on Earth. Joining me today from the Medical University of South Carolina, Michael Schmidt.
Hello, Vincent.
How are you, Michael?
I'm doing well.
As we head into the end of August here.
As we head into the end of August and everybody has to transition out, as my dear Aunt Marie would say, you have to transition out your white purse and your white belt and white shoes for the darker fall colors.
Is that what you do, Michael?
No. No, it's summer always here in Charleston. Though Michele is probably got snow already in Michigan.
Hey. Take it easy.
Also joining us today from Ann Arbor, Michigan, Michele Swanson.
Hello.
Welcome back.
Thank you. Yeah, I'm thinking if I do this once or twice more, you guys might share the secret handshake with me.
So you think we make fun of Ann Arbor? That's why you don't want us talking about snow?
I doubt you'd make fun of Ann Arbor. It's a fine place to live.
I mean, we have so many podcast guests from Ann Arbor, you know.
And this week it's exciting because the undergraduates and the graduate students are moving back.
Yeah.
So we've got, 35,000 people moving into their apartments and dorms. So
what is that like?
Well, yesterday I couldn't swing a cat without hitting mom and dad's SUV,
Wow.
double parked on the street. But it's exciting. We can hear the band playing the Victors, getting ready for our opening football game this weekend. And yeah, it's fun.
Gosh, that's a lot of people. That makes a big change. So the normal population of Ann Arbor is what?
It depends on how big you declare the city, but it's, 110, 115.
That's pretty large.
Yeah.
1,000, not 110 and 100.
Yeah.
Some of our listeners from New York City may think she's actually being literal with only 110 people without the students.
No, I understood what she meant. I brought my son to college last week. He's a freshman, and his college only has 3,000 or 4,000 students.
Wow.
And but you could see all the parents. It's a small campus on the Hudson River. Amazing view of New York City. And it's small, so that we were all crowded in. It looked like a lot of people, but it wasn't really. It's in Hoboken, New Jersey, which is very crowded on its own anyway.
Mm.
Hoboken, by the way, Michele, is the birthplace of Frank Sinatra, your favorite singer.
Have you been reading her Facebook page?
I guess
you've been reading Michele's Facebook page.
I guess you don't like Frank Sinatra, right?
I love Frank Sinatra.
Really?
Sure.
No, you don't have to say that to be on TWiM. I'm just curious. My daughter, who's 15, loves all that era of singing. She can peg a Frank Sinatra song in the first few
all
bars.
That lounge music, huh?
Anyway, he was born there. That's the fame of Hoboken. It's quite a up-and-coming place because it's right across the river from the city, and it's got a very nice community. Anyway, we digress as usual here at the beginning of all of our podcasts. I wanted to, before we move into the science for today, make a little announcement of news, which is a bit old. It's a month old. But our friend Joe Handelsman, who of course is a microbiologist from Yale University and who has been on TWiM a number of times, has been named by President Obama as the Associate Director for Science at the White House Office of Science and Technology Policy. And she's decided to take that. I guess you can't say no to something like that, right?
Nope.
That's right.
And she's going to take that for 2 years and try and make an impact on microbiologists and understanding thereof in the administration science. So congratulations.
I can think of no one better to help American science. She's so deep in terms of what she knows, and it will go beyond microbiology. So
she's also a terrific advocate for education and for science education. So they're they've made a great choice. And hopefully her Senate confirmation hearings will go smoothly.
That would be interesting. I guess, can you watch those?
On
I don't know.
On C-SPAN you can watch everything.
That's right.
You just have to figure out when.
It would be fun to watch because she's so eloquent. It would be really cool to hear her with all the questions. Now, about that nanny you hired in 1992. Anyway, that's really cool. We're not going to have Joe on TWiM for a couple of years, unfortunately. But I think it's a really good thing for her to do, and she can have a big impact. So go for it.
And we knew her when.
Yeah, we did. Okay, we have two cool papers today to talk about, and the first one is a paper recently published in ASM's journal MBio, their open access journal, so everyone can read this paper. It's called Superantigens are critical for Staphylococcus aureus infective endocarditis, sepsis, and acute kidney injury, and the authors are Salgado Pabon, Breshears, Spalding, Merriman, Stach, Horswill, Peterson, and Schlievert. It's from Department of Microbiology at the University of Iowa and the University of Minnesota. And
if I could interrupt, I had the pleasure of talking with the first author yesterday, Dr. Wilmara herself, and I'd like to share just a little bit about her background, if I could.
Love to hear it.
So she was born and raised in Puerto Rico. Her mother was a teacher and her dad worked with the phone company. So her first real exposure to science was as a high school student. She had the fortune of going to a science summer program that focused on both science and math. And that's where she was introduced to microbiology. She remembers very well a project where they were encouraged to find new antibiotics in natural products. And so she had a eureka moment when she found that garlic extract was actually very good at killing bacteria. So that launched her career in microbiology. She then moved to Madison to go to get her undergraduate degree, and then stayed on for both a master's and a PhD. So she started working on bacteria there, studied Neisseria gonorrhea, which some of our listeners will have heard of. And then she did a postdoc with Philippe Sansonetti in Paris at the Pasteur Institute and started studying the immune system and how particular pathogens interact with T cells in the immune system. And then after that, she moved back to the Midwest and joined the lab of Patrick Schlievert, where she's been working on this project. She also is the mother of 3 girls who are now in grade school.
Wow.
So she's got a full, happy life.
T cells, so that is the connection with this paper today, as everyone will see. Well, that's pretty neat.
Yeah.
So did you tell her paper was going to be discussed on TWiM?
I did. She was excited. And she said, actually, there's been a lot of interest both at Iowa and elsewhere in the paper. So, and we can talk more about that as we get more into the story.
So this paper just caught my eye. I look for TWiMable stories every week, and MBio is a great source because they publish really interesting stuff. And we've talked about Staph A bit on TWiM. Didn't you go to a Staph A meeting, Michael?
We went to a Staph A meeting last year, and we reviewed the entire meeting with my good friend Joe John. And
That's right. I think that's the last time we've talked about it. We haven't talked about much disease caused by staph. And so this caught my eye because, as you will see, as you could hear from the title, it's involved in endocarditis, sepsis, and acute kidney injury. So I don't know a great deal about any of those. So I always view doing a paper for TWiM and TWiV and TWIP as a learning experience. I get to read papers that I wouldn't normally read every week. And that is, I tell you, for every scientist, it's probably one of the best things you can do to move out of your field, move out of your comfort zone, and read something that you don't normally work on or think about. And I guarantee you it will give you new ideas about your own work. So we have to do a little background here before we get into the data. And first of all, Staph aureus, second leading cause of bacteremia, and the leading cause of infective endocarditis, and that is inflammation of the heart endothelium, largely the valves of the heart. And these infections have been increasing in recent years due to the greater use of intravascular devices and our increasingly ill population, that is, people with diabetes, liver disease, people on renal dialysis, immunosuppression for various reasons have all contributed to these numbers. Now, often you get a bacteremia, a Staph Aureus bacteremia, when you have a skin infection, for example, or a catheter that becomes infected, a surgical wound, pneumonia, intravenous drug use, and this is associated with a 20 to 40% mortality rate, much of which is a consequence of infective endocarditis. And when you develop infective endocarditis, you get what are called vegetative lesions on the heart valves. And these are meshworks composed of host proteins and bacterial aggregates. And we'll talk more about those in a minute. Now, the crux of this paper is that there has been evidence in recent years that superantigens contribute to infective endocarditis. And there is a recent international study of patients with infective endocarditis which showed that the strains that caused that disease in those patients, a high number of them contained genes in Staph aureus encoding superantigens. And these include staph enterotoxin C, which is the topic of this paper, and a toxin that maybe listeners would know, toxic shock syndrome toxin 1, also a superantigen. Now, in that study, apparently, according to the authors, and taking their word for it because I don't know the field, the high prevalence of these genes have been dismissed by many who say they are simply biomarkers for infective endocarditis, and they're not really causative. So what they've done in this paper is to do some experiments in an animal model to see if, in fact, superantigens play any role in infective endocarditis. Am I okay so far, Michael?
You're okay so far. Are you going to tell us what the magic about superantigens is?
Yeah, I was just going to do that. I know a bit about superantigens because there are a few viral superantigens that are well studied.
I would think so.
And so superantigens are basically microbial proteins that I say short-circuit the immune system. They can be viral, bacterial, as you can tell from this paper, fungal. They're very powerful T-cell mitogens. And what they do is they bind to MHC, major histocompatibility locus, class II molecules on antigen-presenting cells, and then they interact with the T-cell receptor V-beta chain. It's a specific protein chain of the T-cell receptor, which, of course, is on T-cells. And normally, when microbial proteins interact with a T-cell receptor, it's just a fragment of the microbial protein which is presented to the T-cell, and then your immune system makes a response to that epitope, a very limited response, something like 0.001 to 0.01% of your T-cells will respond to an antigen, and then a response to any microbe carrying the antigen or an infected cell to try and eliminate it. In the case of superantigens, the whole protein binds to the T-cell receptor, and instead of getting just a few T-cells activated, you get huge numbers, like 20% of the cells of the T-cells in your body will be activated, and this basically subjugates the immune response. So instead of making a specific T-cell response to a particular peptide, you get this huge amplification of all kinds of T-cell specificities, and so the pathogen basically is using this to its advantage. It's evading the immune response because the T-cells are going haywire, essentially, so they're not really focusing on the pathogen. Plus, they end up making tons and tons of various cytokines, and this causes pathology in itself. That you may have heard of the term cytokine storm, and that's a component of toxic shock syndrome, and this interferes with the adaptive immune response itself. So superantigens are microbes' way of interfering with a coordinated immune response and really diverting host defenses away from the pathogen, and these end up damaging the host. So a couple of genes in Staph aureus encode superantigens, and the purpose of this paper is to find out if that's the case. So in this paper, they use a rabbit model for endocarditis and bacteremia and kidney infection with Staph aureus. And this is very interesting because mice can also be infected with Staph aureus, but they say early on in the paper, murine models do not account for the action of superantigens because mice on a per kilogram basis are greater than 10 to the ninth more resistant to superantigens than humans. So despite the fact that you can infect mice with Staph aureus and it will replicate in them and cause lethality under certain conditions, they don't respond to superantigens. However, rabbits are like humans in their susceptibility to superantigens. So that's very interesting. That's one of the reasons why some animal models are better than others to study human diseases.
And that was one of the stories that came out of the Staph conference last year, is how the rabbit model looking at virulence for Staph aureus is often superior. And, a lot of biologists love to use mice because of their inbred nature, the fact that you have many different backgrounds that you can characterize your experimental question on. And so mice are superior because of their genetics, but they're also inferior because they don't mimic the disease process that we're seeing in people. And some people postulate that was the reason that the Staph aureus vaccine failed, is because, the definition of a vaccine is, do you make antibody? And while many of the vaccine candidates did indeed make antibodies, they unfortunately didn't mimic the response that was seen. And it, quite frankly, was a consequence of looking at it in mice and how they were doing the readout of their particular assay. So I was really glad to see that this group was using the rabbit model to ask their question about superantigens, because I think the adaptive arm of the immune system is often what's missing in some of the pathogenic work. Really trying to ask the question, especially when you have the power of the genetics of the bacteria, where you can do some of the fine structure deletions that they have done in order to ask the binary questions.
So they have picked this rabbit model for this reason, and they infect with a strain of Staph aureus, and they focus on initially two genes encoding superantigens, SEC, Staphylococcal enterotoxin C, and then another one, SELX. And this is amazing. In culture, the bacteria produce 80 to 100 micrograms per milliliter of SEC in liquid culture. That's a lot. Mm-hmm. And the other superantigen, SELX, is much lower, 0.0001 to 0.03 micrograms per mil. So they have strains of bacteria lacking the genes for each of these superantigens. And so the experiment is, in a way, straightforward. You are going to infect the rabbits with either the parental or the mutant strain, and you see the effect on pathogenesis. And the way they do this is they first actually damage the aortic valves of the rabbits. They put a catheter in the carotid artery, and they push it in towards the valve, and they move it around and damage it. And then they inject the rabbit intravenously with bacteria. And I guess that's to mimic, in people who get infective endocarditis, it's often in people with damaged hearts, right?
Or you can actually just get it from an aggressive dental procedure where you actually become bacteremic as a consequence of the vegetative cells that get driven in. Not so much with staph, but with the strep, specifically strep sanguinus, or one of these others that can result in vegetative endocarditis.
And does that happen in people with normal hearts, or do they have some damage?
Normally, your surveillance system is sufficient. Typically, where you see the vegetative endocarditis as a risk factor is in people who have had rheumatic disease, where their hearts have been damaged as a consequence of, the immune system cross-reacting to one of the streptococcal antigens that looks like one of our heart antigens, and that's why it's called a rheumatic disease. It's effectively a consequence of your immune system doing what it's supposed to do.
Right.
And Myra did explain in their rabbit model, it's the same scenario. So if they don't damage the heart valve, they won't get colonization of those valves. So the staph really does need to bind to the damaged tissue. She described that it takes 4 to 6 of them all coordinating their efforts to do these experiments, to do these surgeries that are very, you have to be very careful because, of course, you're keeping the rabbit alive during the course of the infection.
I can imagine that it's tricky to just do the right amount of damage to the valve and not too much, right?
Right. So they have this whole team, an assembly line of 4 to 6 people, do each with a specialization in the procedures. So it was a neat image.
Yeah, I was lucky to have had visited this department in February, and I talked with the graduate students. So I heard about this production line, as Michele said, and this is. You learn collaboration about research very early on in your careers, and I think this team that's working on this model should be commended because it is a finesse technique, but it's really what's happening in people when they get this devastating disease, and we really need to understand it in order to control it.
Yeah. So in their first experiments, they infect the rabbits intravenously and then wait 4 days. And in this time, the parental strain, which is called MW2, causes lethal sepsis in 80% of the rabbits. So there's a disseminated, virus disseminated in all parts of the rabbit, and 80%
bacteria.
Sorry.
We're listening.
I'm so used to saying virus. Bacteria disseminated, 80% of the rabbits died. And then the sec deletion, the deletion of the sec gene, that's again staph enterotoxin C, the superantigen. 90% of the rabbits survived to day 4. And the deletion of the other superantigen, SELX, had no effect on lethality. So that's the last, actually, we're going to see of that gene in this paper. So clearly, SEC is a dominant virulence factor, at least in this assay, which is really just for lethal sepsis so far. An interesting point here is that the counts of bacteria in the blood are not different between the parental and the mutant strains. So, the attenuated lethality of the deletion is not due to having simply fewer bacteria in the blood. And that's always an important experiment. Even with viruses, we have to make sure that when you delete a gene, you don't just knock out replication, because that isn't very interesting, obviously. Then they look at the infective endocarditis, and they infect these rabbits with the damaged aortic valves, and they wait longer to see the development of these growths on the valves. And they say, which I think is a very interesting way to describe it, the parental strain MW2 formed large vegetations with an off-white cauliflower-like appearance. So you have a lot of vegetable analogies here. I wonder why. And they describe these vegetations, they're composed of blood clots and bacteria and host tissues on various valves. And the rabbits that die have lots of vegetation. So here in one experiment, 9 out of 9 rabbits infected with the wild type developed vegetations, and they weigh them, and they can see how big they are and how many bacteria that they contain. But of the 10 rabbits infected with the Delta sec strain, 7 had no vegetations, and 3 had small ones that weighed much less than the vegetations in the wild-type infected animals, and they had fewer bacteria in them. So apparently this superantigen also is influencing the development of vegetations on the heart valves.
And it's quite striking.
It is. The difference is striking.
Yeah.
Did she give you any insight into this?
She did. She described, I asked if was there a particular day in this project that was especially memorable, and she said it was when she opened up the animals, and she had done the wild type and saw all the usual cauliflower lesions, and then opened up the mutant. And, in her head, she was thinking she'd still see some and maybe they'd be a little smaller. But she looked and looked and looked, and they're just gone. So it was a very exciting time at the bench.
So again, remember that the bacteria in the blood is not changed by the deletion of the SEC gene, but it's simply the formation of these vegetations. I don't know where it got the name vegetation, but it's the food aspect of it is kind of off-putting.
It's because they grow on the tissue.
I know, but cauliflower is really pushing it.
Well, it's descriptive. This was before the days when we had digital cameras in our pockets.
Yeah, it's okay. I'm just complaining, but it's fine. I don't really have a problem with it. The next thing they looked at was kidney injury, because that is one of the effects of SA infections in people. You get kidney damage. So they infect their rabbits again, and they remove kidneys from rabbits infected with the wild type or the mutant strain. And again, they see a big difference. Lots of abscesses and sites of damage in the kidneys of rabbits infected with the wild type strain, but the majority, 7 out of 10 infected with the mutant strain lacking the SEC gene, had normal kidneys, no ischemic tissue, that is lacking in oxygen, and no abscesses. So apparently this superantigen gene is also contributing to kidney injury as well. They go on to characterize these vegetative lesions quite a bit, because you really would like to know how they're formed. What is it about a superantigen that induces them? And again, these are in rabbits infected with the parental strain. They have vegetative lesions. They're made up of fibrin aggregates. They have platelets and erythrocytes in them, and bacteria embedded within the aggregates of these proteins. And so they're wondering if, the massive cytokine production is somehow playing a role in the development of these. You can imagine that might damage tissues. They also wonder if the superantigens are directly affecting the endothelial cells of the aorta, which would lead to this as well. So they get an idea initially of what these vegetations look like. Now, the next experiment is pretty interesting, and this involves fluid replacement in the rabbits. So they have a hypothesis that as the bacteria are making superantigens in the heart area, for example, it's causing the capillaries to leak, because remember, the superantigens our immune cells make lots of cytokines, and they can cause the capillaries to leak. And this causes reduced blood pressure or hypotension. And they have a hypothesis that the hypotension could allow the bacteria to settle on the heart valve. So if you don't have enough intra-arterial pressure, it could allow the bacteria to more readily settle onto the damaged valve. So they infect their rabbits as usual, but then they inject them with every day or twice a day 25 milliliters of saline. And so this is to restore the blood tension, if you will, the fluid loss accompanying the infection. And they actually find that the fluid-treated rabbits have smaller vegetations on their heart valves, and they have fewer bacteria in the vegetations compared with rabbits. Again, these are infected rabbits. These are with the wild type or the parental bacteria that didn't get the PBS. So just giving them fluid significantly helps, which is pretty interesting, because that's something you could do in people.
Right.
Yeah.
Right, directly. And I can imagine a clinical trial, because giving people PBS is not a problem, I would guess.
No, and in fact, you, as the learned clinical professionals in the infectious disease groups begin to look at this primary research, I mean, it's a standard of care of, just managing fluids as part of the patient's care. And, they could do it. But true full-blown informed consent, where, you will place, if this is true in humans, you won't ethically be able to do a clinical trial, because you'll be placing the control arm, the one that doesn't get the fluid, at a greater risk.
You can't withhold IV fluids, right?
Right.
Yeah.
When their blood pressure drops due
That's
to the
true.
leaks. Yeah.
And in fact, that's one of the ways they manage endocarditis is with pressors to keep the pressure up. And, something as simple as flow. At first, when I was reading this, before I looked at all the controls, I was concerned, well, the solution to pollution is dilution, and they may be just diluting out the cytokines that are being produced locally. But then with their controls, I came around to believing that it was indeed a pressure effect, and by replacing the volume to keep the pressure up, you could do it. But another appropriate control is you may be able to increase the pressure in these animals and be able to ask the question whether that will ameliorate the endocarditis.
So there's actually two different things going on here. The first is you have a bacteremia, and then these bacteria are producing superantigens that's reducing your blood pressure, and that helps to colonize your aortic valves, right? So the question is, if you in an animal model, if you raise the pressure, well, that's what they did here. They raised the pressure before or immediately after infection. That prevents colonization. Mm-hmm. But in a person that's already colonized, say, who presents with endocarditis already, would increasing the pressure help? In that case, it might be an effect of dilution, because if the heart valves are already colonized, then pressure wouldn't apparently make a difference, right?
Yeah. Well, that's, one of the hallmarks of managing endocarditis is they always make the argument you must use bactericidal antibiotics as opposed to the static ones. You truly want the vegetative cells to be dead as opposed to just sitting there in a stasis mode. And it goes back to your original opening line of the superantigen effect. Activating 1 out of 5 T cells versus 1 in 10,000 T cells, it's the cytokines that are responsible for the coincident damage of the lining, the endothelial linings, in both the heart and then the kidney. And, once you begin to go down the path of organ failure, it's a very slippery slope to reverse that process. And so, beginning to tease this apart in this very straightforward manner with these two mutants will really give us, I think, tremendous insight into understanding the whole issue of endocarditis. And remember, staph is good friends with its other endocarditis-causing microbe, strep. And, strep makes toxic shock-like toxins as well. And, we've talked about toxins before, and there's an A and B component of each toxin. And, if we can understand how these things are working, we may be able to come up with a therapy that will, in concert with appropriate antibiotics that are cidal, will actually have a good outcome for the patient.
Right. So the last experiment, they test another of their hypotheses. So far we think that a lot of the damage is caused by cytokines produced by this immune response gone crazy, the superantigen stimulation of T cell proliferation. But they also wonder if the superantigens could work directly on the endothelial cells, the cells that make up the vessels. So they have human aortic endothelial cells in culture, and they simply treat them with purified staph endotoxin C. And they measure a cytokine in the supernatant just as a measure of whether the superantigen's turning on cytokine production. They measure IL-8, interleukin-8. And they find, in fact, that the addition of the superantigen stimulates IL-8 secretion from these cells. And the production of IL-8 is known to occur by a certain pathway. And they know that other superantigens, in particular TSST1, taps into a signaling pathway in the cell. So they just simply use inhibitors of this pathway, and they show that things like metalloproteinases and other VEGF receptors, for example, inhibiting those interfere with the production of IL-8 in response to the superantigen. So this is actually news to me. I didn't know that superantigens could have direct effects on non-immune cells, non-T cells. So this is pretty interesting too, because the superantigens could be damaging or could be causing the endothelial cells near the valves to produce cytokines, which in turn are damaging. They damage the tissue, and that exacerbates the whole thing.
Right. And gives the bacteria more places to colonize.
Exactly.
What's exciting about that is because it's not the whole bacterium, but rather this superantigen, that opens up the possibility of vaccine development.
Right.
Or, as you mentioned at the beginning, Vincent, since the superantigen is known to bind a particular part of a T cell receptor, this same group has done experiments in rabbits showing that if they inject the rabbit with just the fragment of the T cell receptor that is known to bind the superantigen, it can basically act as a decoy and dampen the effect. So inactivate the toxin by binding it and keeping it off the endothelial cells, for example.
Yeah, that's a cool way. You can imagine making a small molecule that does a similar thing.
Or better yet, a filter. All you need to do is attach these small molecules into a filter, and then you just passage the blood through it. And the toxin, since it's diffusible, as you run the blood through the filter, will effectively bind up the toxin and effectively starve the bacteria or prevent the bacteria from finding new niches from the cytokines or these toxins being sequestered. And, a lot of folks are beginning to think about the path to licensure is much easier if it goes on outside of the body rather than inside of the body.
Interesting.
It is very interesting. I hadn't thought of that, yeah. So there you have it. It's quite an interesting paper. It takes this suggestion that superantigens are important for infective endocarditis, kidney damage, and septic lethality, and they actually show it in a rabbit model. And I like their last sentence. With this knowledge, it becomes possible that patients may be treated with therapeutic agents that neutralize superantigens, limit disease progression, and thereby increase survival. So I thought that was really nice. It was a good paper.
And remember, one of the other enterotoxins or exotoxins of Staph aureus is its enterotoxin. That's responsible for the projectile vomiting, often associated with staphylococcal food poisoning. And that's also a superantigen-mediated consequence. And so, again, as you begin to think about markets for your particular low molecular weight compounds that you want to make, just imagine if you could somehow develop if you do develop the symptoms of staphylococcal food poisoning, which occur very quickly, and you could have an over-the-counter medication that you could keep in your medicine cabinet that you could take if you had the symptoms of projectile vomiting and the classic staphylococcal food poisoning symptoms, you could feel better before, staphylococcal food poisoning is typically self-limiting, but you generally feel extremely hungover after the intoxication has taken place.
And just to emphasize how powerful these superantigens are, some of our listeners who are also Frank Sinatra fans might remember the outbreak in 1978. It started. Healthy young girls, teenage girls, were being admitted to hospital with shock. So that was actually the first description of toxic shock syndrome. And it was then traced to these super absorbent tampons called Rely tampons that were made with a synthetic gel-like substance that was actually a very good medium for promoting Staph aureus in the vaginal cavity. And so healthy young girls were getting toxic shock syndrome, and some of them died. So that just shows how potent these superantigen toxins can be, and how staph, which is really very common on our body and on our mucous membranes, if given the opportunity, can really wreak havoc in short order.
And from that original incident came the identification of TSS1, right? And
actually that was the first description of toxic shock syndrome.
Huh.
And thankfully, now that we understand more about tampon construction, we're no longer using those synthetics that put young women at risk. And so the incidence has dropped once they took the Rely tampons off the market. The incidence and education also really dropped the risk to young girls.
And the principal presentation was hypotension, low
Right.
blood pressure. And again, it drives home this theme of what these things are doing. And this is why this is a very interesting paper to read, from both how they approach asking the question, answering the question, and then actually teaching you something about staphylococcal pathogenesis. So if you're new to the field, I commend, as our good friend Elio would say, I commend this paper to you because I think you'll actually enjoy it and get something out of it. And again, one of the hallmarks of MBio papers is that as you begin to go through the figures, you really impressed it at the rigor that the editorial group makes these authors adhere to, because you can figure out the figures.
Our next paper is also very informative, and you will also enjoy, but it is published by Cell, so you may not be able to access it, unfortunately. It's all about the axillary microbiome, and Michael would love to tell us about this.
Well, I was looking for a paper for the end of the summer, because it's a daily battle against sweat and body odor. And
Speak for yourself, Michael.
Well, you're not here in the swamp. I'm here in beautiful Charleston, South Carolina, which 9 months out of the year is Chamber of Commerce weather, and for 3 months, from probably the end of June until the end of September, it's quite humid and quite hot. And so consequently, you appreciate what the microbes are doing. But I really picked this paper because I think it's going to be a primer for us to think about how we're going to begin to dissect the complexities of the microbiome. The human axilla is not a very complicated niche from the perspective of a microbiome. It's not a very...
Tell us what an axilla is, Michael. You're using your fancy terms.
Oh, well, let me go less fancy and just refer to it as it's the underneath your arm, or more affectionately referred to as the armpit.
You know, some people call New Jersey the armpit of the nation.
Yeah, I wasn't going to go there, Vincent. You're a citizen of the state, and you're going to get a letter from your governor.
You know, the governor actually visited my hometown the other day. I was telling people here that we were going to do this paper, and that they actually made the statement that New Jersey is the armpit. But in fact, I just want to point out that it is not, because we have some very beautiful areas of New Jersey.
It's, I've been to New Jersey many times, and its license plates that refer to it as the Garden State. And if you actually drive through the back roads, it is really pretty.
I feel compelled to just take 30 seconds to defend the state. For the last, we have a farmer's market in the neighboring town, and all summer, every Friday, we pick up a box of produce made in New Jersey, a selection of Jersey stuff, corn and tomatoes and cukes and peppers and eggplants, all local stuff. It is just amazing what's grown here until October, November even.
And do you wash it well?
I don't really wash it very well. I have never been a big fan of, I rinse it, but that's nothing, right? Actually, one of our listeners, and it's probably not queued up, sent us a way of washing vegetables in dilute vinegar, which apparently is very good. Anyway, I don't wash it all that much, but I've never been sick.
Yeah, your microbiome is taking care of you. So this hollow beneath the junction of the arm and the shoulder, or the armpit, is the human axilla. And we all know from the time we reach puberty that we begin to smell differently. And this is really, the smell is an exercise in a consequence of what's eating us. The microbes. when I go out and talk to middle school children and when I go out and talk to high school kids, I try to introduce microbes from a perspective of something that they can actually immediately relate to. So I normally ask them to take their little finger and rub it behind their ear and smell. And, the skin is a rather remarkable organ of our body. And, behind your ear is a moist area, but it's typically dry. And you can actually smell those volatile compounds that are coming out. And I typically describe those volatile compounds to the high school kids as short-chain volatile fatty acids. And to the middle school children, I say these are natural chemicals that are coming off of our bodies that the normal flora of your skin are transforming, and that's effectively an indication that they're eating the waste products that you're actually putting out as a consequence of sweat. And as we hit puberty, our sebaceous glands begin to move other steroid-like molecules, because as we go through puberty, we begin to dump more steroids into our system, and consequently, they too are secreted. And we have two other fancy words that we have to introduce, and I'll start with the simple words. Principally, the sweat glands, or the eccrine glands. They're responsible for secreting clear and typically odorless substances, and they release mainly water and electrolytes like sodium, potassium, calcium, magnesium, chloride, bicarb. And then they secrete something that bacteria, specifically the ones in this particular niche, can actually make use of. They secrete lactate, urea, and ammonia. And appreciate the fact that all bacteria need a carbon source. In this particular case of the sweat gland, it's going to eat the lactate, and they need a nitrogen source. And so that could either be the urea or the ammonia. And then, the other molecules of life that you're going to need you're going to need some sulfur in order to get along. And they typically get that from amino acids that are being secreted by our systems as well.
What about iron, Michael?
The iron, they're going to scavenge from the rest of you. You're probably secreting a trace concentration, but I don't know how much is actually weeping away. Our bodies are pretty stingy with iron. We lock it up in a box called transferrin, and we're very stingy with letting it go. And then the apocrine glands, the so-called scent glands, actually dump out high-density nutrients that are highly characteristic of that axial region for human adults. And, this is a milky-like fluid that, it's very difficult to get in quantity from people. And so consequently, that's where you get your lipid fraction and the other things necessary and sufficient to grow more bacteria. Now, most of this work has been underwritten by the cosmetic industry, which makes perfect sense because the cosmetic industry has been very much interested in understanding body odor formation mechanisms so that they can then begin to design inhibitors to particularly mask the odors and to particularly control the way we smell. And the cosmetic industry, I think, is ahead of everyone in this field of trying to understand how to exploit the microbiome. And that's effectively why I picked this review paper that was in Trends in Microbiology, because I think it can serve as a primer to help those of us interested in looking at the extremely complex gut or the extremely complex oral cavity or these other niches of the body that are especially important in medicine, that I think it will begin to teach us what we need to do. Because previously, the way the cosmetic industry has controlled body odor and sweat is they've actually used harsh chemicals that have actually first disinfected that area or killed off the natural flora. And, if you do long-term scorched earth management of anything, it can become an irritant. And we really have to try to figure these things out. And so that's the second aspect of their paper, is they describe the origin and composition of this human axillary body odor. And they go through the compounds, and they describe the microbe and the enzyme responsible for the transformation of the precursor that's literally coming off of us. So, for example, one of the things that is being secreted as an odor precursor is something called glycylcystenyl conjugate. And anything with sulfur in it can be picked up by our noses when it's transformed into the active ingredient, which is a 3-methyl 3-sulfonyl hexane with an alcohol moiety. And mammals are really good at picking up low molecular weight compounds that have a sulfhydryl in it, because hydrogen sulfide, of course, is lethal to air-breathing animals at a very low concentration. So it's probably hardwired into our basic DNA to avoid anything that resembles hydrogen sulfide. So our odor detectors have effectively retained that phenotype throughout evolution, because, if you're too close to a volcano, you will die. And in the case of this glycylcystenyl conjugate, it's a Corynebacterium and a Staph Aureus species that actually have dipeptidases and lyases that transform it into this active odor compound. And they did an interesting piece of biology characterizing people, looking at which microbe was responsible for the production of which odor-causing compound. And they blamed the poor Corynebacteria simply because of how they began to ask and answer the question, could staph, and the staph that they've actually named, and then the damn taxonomists get involved, and they change Staphylococcus asaprophyticus to another thing that became pepto something or other. It loves peptone, and they began to change the names of the microbes, and they go through the very familiar technique of looking at the microbiome from volunteers, specifically isolated from these areas, and they do 16S ribosomal DNA sequencing to begin, and then they move over to RNA sequencing, making the argument that they want to understand who's actually alive. I mean, one of the hallmark problems associated with characterizing sequences is you don't know whether or not the organisms are alive or dead, because it's just measuring nucleic acid, and you're amplifying it up via the polymerase chain reaction, and so you don't really know who's contributing. Whereas RNA, because of its extreme short half-life in the microbe, is, of course, a hallmark of who's actively growing under a particular circumstance. One of the interesting things about their description of how odors come into being is they look at the branch-chained amino acids. And again, our friends Corynebacterium and Staph Aureus are following along. But then some of these low molecular weight compounds like lactic acid and glycerol, they bring up this fermentation pathway, the Wood-Workman cycle, and this should cause many of us who remember our microbial physiology fondly, this should cause us to smile, but it will also cause other graduate students out in the audience to break into flop sweat during their qualifying exams when they got sent to the board to diagram the Wood-Workman cycle. And, of course, this is where Propionibacterium comes into vogue. And when I teach the dental students about the Propionibacterium, of course, Propionibacterium is the causative agent that causes acne. And I asked the class, how many of you have ever popped a zit? And I tell them that all of you have to raise your hands, because there's not one individual in here that did not pop a zit right before prom. And they, of course, look at me like I'm half crazed, but they immediately remember. And I said, how many of you actually looked at the mirror and got your fingers very close to your nose as you began to pop that zit? And if you recall the smell that was revealed when you popped the zit, it had the distinct odor of Swiss cheese, which is, of course, the principal organism responsible for Swiss cheese will produce a large amount of propionic acid and CO2, which puts the delightful holes in Swiss cheese. And consequently, they begin to immediately appreciate that you can identify organisms on the basis of smell. So they review a lot of work in this review article that came out in June, and it's just a coincidence that as I was going through reading things, PLOS One came out with a paper just this week actually covering this very topic on the Human Axilla, and they did a additional sequencing project looking at this. And so they go through this and they begin to look at the microbes that are associated with the human axilla, and they go through the genera, and the usual suspects are there, the Crinibacteria and the Lactobacillus and the Propionibacteria. But they bring up the caveat because they initially began to use Sanger sequencing, and they thought that that may bias it. And so they, again, bring up all the limitations, rightly so, associated with some of the microbiome literature that is emerging. So again, from that perspective, this review is good because it begins to teach you to begin to think about the limitations of the studies that you may or may not be reading. And they have very colorful figures. And the second figure actually describes in detail a number of studies from healthy volunteers and the amplicons that they look at and the methodology and the differences that they actually see. And they're using colors that allow you to see who's there and allow you to assess for the differences and complexities.
There's pretty big differences between individuals.
Oh yeah. And that's one of the hallmarks. Is
this something you would find in the other skin, or is this unique to the axilla?
Well, the axilla, and I've given our good friend Ray a note, is a very moist environment. So
speak for yourself.
It's a moist environment from the perspective of your back,
sure,
where it's much drier, or your legs where it's exposed to the air and it's much drier. And ASM did this wonderful television series that aired on PBS as part of our centennial in 1999 called Intimate Strangers, Unseen Life on Earth. And those videos are available on iTunes. And the Dangerous Friends and Friendly Enemies opening montage has this man and woman doing the tango. And it actually zooms in on her axilla. And it actually shows you the rainforest of microbes that are growing in the human axilla. And so I didn't find it all that surprising that there was inter-individual variation. But what I did find remarkable is that as they began to move from DNA to RNA, they identified higher intra-individual variability based on the RNA, And they were able to learn that there was a higher activity of this pepnonephilus, which used to be a staph, and it was based on which arm had the higher abundance as to whether or not you were right-handed or left-handed, which I found as a fascinating coincidence that the microbes could actually tell which hand you were using more than the other. But I guess it makes intuitive sense because you'd be moving that hand more based on whether you're right-handed or left-handed. So they're asking for a more focused experimental setup could yield deeper insights not only into the diversity and variation of the axillary microbiota, but also into the physiological functions, which are differentially expressed by the metabolically active members of the microbial community.
So basically, these differences between people would explain why some people have more body odor than others, I presume.
Yeah,
or different scents. Let's say different scents.
Sure, but you will agree, Michele, that some people do not smell at all, and others smell quite a bit, and then others have different kinds of smell. I mean, my kids are always complaining about certain individuals in school who really smell, and others do not. So there's a big range, and I guess you could do studies to probe that, right? Really see.
And in fact, that's how they tease us at the end. One of the things I love about trends in microbiology is their outstanding questions figure that they always seem to end their papers with. And the first one that they ask, is it possible to establish a standardized protocol for sampling and metatranscriptomics of the axillary microbiota? And their third figure actually shows you how they are advocating a standard sampling. And if you begin to think about some of the microbiome papers where we've talked about stool and other complex environments, that's what the FDA is struggling with presently, is how do you qualify a stool donor and all of these other things. And what they're arguing is that you do total RNA isolation and then you do the cDNA amplification. But they rightly point out that the bacterial and archaeal messages don't have the gift that the eukaryotic messages have given us, namely poly A, where we can cheat and use poly A T column to recover all the message. We sort of have to, use adapters and amplify everything and hope for the best. And because messages are so unstable in the bacterial world, where the average half-life is between 20 seconds and 2 minutes, you can begin to understand how you have to really swab the armpit and drop that into the guanidine isothiocyanate and hope for the best.
Michael, I could imagine that axillary hair would also make a big difference. So I wonder if in these studies, just typically men tend to have more frequently. They don't shave. And more women tend to. So I wonder if the study was adjusted in any way, or because
they used men. They used men for one of the studies, if I'm remembering properly, and that's the problem. The way I refer to these review papers sometimes is they're mashups, and you almost have to go back to the primary literature. But I thought this was we tend not to do reviews because there's so much data in them and there's so many papers. But I really wanted to pick this paper because I think it will lay out for those people interested in understanding how to begin to dissect the microbiome and design good experiments like we saw with the last paper, how we can begin to take the story apart. And, they end their story by talking about the cosmetics industry and looking at how to add candidate deodorant additives that can actually effectively serve as decoys. If you could add an additive that would change the balance, and I think this is truly how we're going to be able to figure out, can we influence the microbiome long term by making small molecules that could actually grow more of a good bacterium versus a bad bacterium. And in this case, we have an operational definition by simply looking for odor. And the analytics folks have gotten really good with gas chromatograms that can actually smell. There's a whole subdiscipline of microbiology just looking for short-chain fatty acids, and that work was originally pioneered by David White, the late David White, who looked at fatty acid profiling for anaerobes, and so looking for those end products. And so we have a way of quickly screening to see whether or not these additives, and it would probably be fairly inexpensive to do in a long-term study where you could add these additives to a carrier and a deodorant. You would apply it into a large group of people, and then you would stick a probe under their arm and smell, and you could ascertain very quickly by looking at the readout whether or not it worked. And I'm sure our friends at Procter & Gamble and the other large manufacturers of deodorant are actually, doing some of these studies.
So in our lifetime, Michael, are we going to see personalized deodorant?
I think we'll actually see it because, if you ask a person, of humans, which humans smell the best, people will say newborn babies. Newborn babies have that fresh baby smell, and that's hardwired into our DNA as well. We all love the smell of small children. We do. I mean, it's you laugh, but all of this stuff is hardwired into our DNA because we take care of our young, and you have to begin to understand what's going on. And this whole issue of these apocrine secretions, where you're putting out hormones and lipids that are being digested by microbes, it doesn't take much of a stretch to begin to move into the next area. And what we are seeing in this week's Nature that just came out is looking at the complexity of the human gut, asking the question, the less complex you are, the greater the likelihood you are going to be obese. And, how are you going to begin to develop probiotics to alter this in a long-term relationship? I think, we have to start simple, and, it's feeding studies will be expensive, but underarm deodorant additives where you could measure some of these cystinyl residues, and, their table gives you all the molecules to check from. And you don't have to be a student of analytical chemistry to appreciate that you can smell the difference between those compounds.
I would guess that trying to bias or change your axillary microbiome could be a problem, because I assume that part of it is beneficial, and
Oh yeah,
there could be problems long term by changing it so that you just smell good.
Yeah.
So that has to be taken into account, but it's often one of those things that we don't find out till years later.
That's right.
Right.
There was a comment they made that struck me. We've been focusing on the diversity of the 9 different people they looked at, or however many that was. But they make the point that if you, instead of thinking about the different species, look at the metabolic pathways, that's actually quite stable.
That is true.
Yeah.
And, I'm glad you brought that up, Michele. That was the other reason I wanted to pick this paper, because we often have gone down the path, because the 16S ribosomal database is so robust. Maybe we need to begin to look at the sentinel enzymes for the Wood-Workman pathway, or these other clearly remarkable microbial physiology pathways. And this goes to my good friend Amy Vollmer at Swarthmore, who's advocating that we must teach more microbial phys. We have to begin to appreciate that we're going to have to go after some other sentinel enzymes other than 16S ribosomal RNA sequences. We may need to look at some of these other key choke point enzymes that determine which pathway you have. And I think that's the way we're going to be able to begin to dissect the sentinel species that can influence whether or not a probiotic will have a long-term effect. And that's, the holy grail of the microbiome is we really want to have inexpensive medicine that we can personalize. And right now they're doing it with brute force sequencing. And this paper may give us a handle on how to think about doing it in a different or more clever manner.
This paper is from the University of Bielefeld in Germany, and the authors are Friedrich Barzani, Brun, and Tauk. And I think this is really interesting. So if you can get a hold of it, by all means, read it. It's a nice article. Thank you, Michael.
Thank you. It was fun.
Let's read a couple of emails here. The first is from Hugh, who writes, I really enjoyed hearing about Carl Woese in TWiM number 50. You mentioned the controversy surrounding Woe's 1977 discovery of archaea as a third domain of life, as it contradicted entrenched scientific beliefs. Although archaea soon found its way into general scientific thinking, his work brings up another fundamental idea that is still controversial today. It relates to the word prokaryote. Norm Pace, one of Woese's former students, is a strong believer that we need to stop using this word. I think he makes extremely compelling arguments about the harm this word does to basic biological understanding, and thought this could be an interesting discussion point on TWiM. Here are the titles of two papers by Norm Pace that discuss these issues. They're both geared to a general audience. Problems with Prokaryote in the Journal of Bacteriology, and Time for a Change in Nature. I recently discovered TWiM and TWiV and am now an avid listener, mostly while cooking dinner and washing dishes, which always seems like deja vu after a long day of experiments and washing glassware at the lab. Thank you for making that process infinitely more enjoyable. Writing from Seattle, Washington. I think a long time ago we briefly talked about this.
Yeah.
I know you remember, Michael?
Yeah, we did. We talked about Pace's getting rid of the word prokaryote and just going to bacteria. I'm trying to remember which episode.
Yes.
We're now up to 63. So,
how do you stand on this, Michele?
I don't have strong feelings about it.
Yeah. As I think Elio was in the same camp. He said, it doesn't really matter.
It's because, you're not wedded to the taxonomy.
Right.
I'm more of what are the microbes doing sort of person rather than hung up, what do we call them?
Yeah. I think it would be interesting if we got one person who was for and one was against prokaryote and had them discuss it on TWiM, but I don't know how you'd do that.
Well, prokaryote just simply means without nucleus. That's all it means. So.
Yeah, I stopped using it a long time ago. So,
yeah.
But anyway. The next one's from Megan, who writes, Hi, TWiM team. Could you do a TWiM about the archaea and viruses that infect them? It's a fascinating area that doesn't get as much attention as bacteria. So she doesn't use the P word. See that?
Yeah.
Yeah, that would be a cool topic. We'll look for papers, Megan. Jim writes, another update on the hand washing issue. This link discusses the article, which costs $10 to see at the Journal. I'm so sorry about that, Jim. He links to a CBS News report entitled 95% of people wash their hands improperly, and they basically describe the study, which was published in the Journal of Environmental Health in June. We can put the link to that in the show notes for those of you who can get to it. They talk about the right way to wash your hands for 20 seconds minimum.
Well, it's really interesting. I was at a seminar last week, and Dave Weber from UNC was giving the talk, and Dave Weber is a noted infectious disease specialist who's a strong advocate for hand washing, and he sits on his institution's or his hospital's infection control committee, and everyone who gets paid by the hospital, 1% of their salary is at risk based on the hand washing percentile that the collective of the hospital achieves. So
interesting.
So it's effectively peer pressure at its best. If you want your 1% bonus, you make certain that your colleague next to you in the toilet is washing their hands properly, or someone coming out of a patient's room has washed their hands properly, because they have employed the secret shopper to effectively ascertain the compliance ratio.
Huh.
And if everybody is above 95%, the entire hospital gets 1% bonus.
Do they have data yet on how effective that policy has been in spread of nosocomial infection?
Dr. Weber is part of a multi-center trial. I don't know specifically it's about the bonus issue as being one of their variables, but he just shared that in passing. But they're, from what I understand, at my own institution, this is, since I'm not paid by the hospital, I didn't know this until I was talking afterwards to our hospital epidemiologist. All of our house staff, which are the residents, 1% of their salary is also at risk based on hand hygiene compliance.
It's a good motivator.
Yeah, well, 1% is real money.
Well, maybe for you, Michael.
For me, it is.
My father was a surgeon, and he would wash his hands at home the same way he did while scrubbing. I've never seen anything like it. But I didn't inherit his ferocious washing. I'm not very good. When I wash my hands, I always say, okay, I'm supposed to recite the alphabet, and I get to about C or D, and that's it. Just like my vegetable washing.
Yeah.
This next one is from Mark. This is quite an interesting. This is extended, so we'll make this the last one. Mark writes, hello, Vincent, Michael, Elio. I enjoy TWiM and have listened since episode number 1. Keep up the good work and keep going. Out of TWiV, TWIP, and TWiM, I discuss episodes of TWiM with my wife the most. I am writing belatedly to correct some inaccuracies in episode 49, grape-like clusters. These are 1. Someone, VR, commented that French wines were the best.
Uh-oh.
This is not fact, but a matter of taste. The Latin expression degustibus non est disputandum applies.
You're in trouble, Vincent.
I did say that.
You did say that. But you were hoping the Californians would send you a case.
I know California wine is really good, but how can you beat a 50-year-old Bordeaux? There's just no way.
I myself have never had a 50-year-old Bordeaux. I'm the more of the two-butt chuck kind of person.
I haven't either, but I can imagine. I understand it's a matter of taste, so I won't extend it, but I think French wines are really good. And so are Italian wines. 2, Michael speculated that natural yeast was used to ferment wine. This is a common belief and is mostly wrong. There is a vigorous industry dedicated to selling commercial yeasts to winemakers. A small number of boutique wineries are experimenting with using natural yeast. These are a minority, and the quality of their results is highly variable. I am an amateur winemaker of 17 years. See the attached image of a macro bin containing half a ton of Cabernet Franc grapes from 2012. Note the white powder-like color on the grapes. This is natural yeast that grows in the vineyard. Note also the stems with missing grapes. As harvest occurs, the growers typically take weekly samples to measure sugar content. In winemaking, after the crush, in which a machine separates the stems from the grape berries, and in which the berries are lightly crushed, potassium metabisulfite is added to the must, the residual grapes and juice, to kill natural yeasts and inhibit fermentation. Typically 24 hours later, the must is inoculated with a yeast starter and nutrients. The image of nubile virgins stamping barefoot on grapes to make wine is sadly a myth.
Well, let me tell you where I got the natural crush from. When I was a poor, starving postdoc living at Stony Brook on Long Island, I lived in the basement of a Portuguese family that used to go out and get 3 or 4 pickup trucks worth of grapes from the Pindar Winery, which is at the end of Long Island. And then they would come back to the house and they proceed to make enough wine for a year. And every time I paid my rent, I got a gallon. And there was no starter culture involved. And it was damn fine wine. At least you could drink it. And so every time you'd give him the rent check, you got a gallon that would, take you through the month. Yeah. And he made red and he made white. And so it was a natural crush. But, the Pindar Winery, it grows incredible varietals. And they make really fine wine. And but my landlord, he made wine. And he did all the crushing and fermentation in the basement below my bedroom. And I would have, every September, I'd have a fruit fly infestation that the first time it happened, I didn't know where all these flies were coming from, but it was from underneath. And unbeknownst to me, when I moved in, I didn't know he made all this wine. And when he found out I was a microbiologist, it was wonderful. But he was a cement contractor, so you didn't cross him.
Okay, he's continuing. Our winemaking group purchases yeast from a company which supplies commercial wineries, Gusmer Enterprises. Attached is their 2012 catalog. Its front and back covers commemorate scientists whose discoveries were key to winemaking: Pasteur, Leeuwenhoek, Riley, or Dewar. And I've attached a link to that here in the show notes. There are subterranean links to virology. Vincent can speak to physicist turned virologist Max Delbruck in his seminal work. Did you know that his uncle, also named Max, developed a yeast, Torulaspora delbrueckii, that will live in high alcohol levels? These yeasts are especially important in California because our climate produces grapes with very high sugar levels, which through fermentation produce high alcohol levels. I didn't know that.
I didn't either. This is really incredible.
I know Max Delbruck, the virologist, of course, the physicist turned virologist who developed the one-step growth curve, the originator of the phage school. 3, the paper that Michael presented provided a quantitative framework to characterize and measure the differences between grapes grown in different blocks on the research vineyard. As winemakers, we see a qualitative difference in each barrel of wine. Rule of thumb, half a ton of grapes equals 1 barrel, equals 24 to 25 cases of wine. It is too labor-intensive to produce and label at such levels. Thus, wine from each barrel is blended, which averages out the different tastes of each barrel. That's cool. I didn't know that.
I didn't either.
You remember that paper, Michael? They were sampling from different blocks, so that's way above the barrel level, which you would, if you did that, you'd see variation as well, right?
Absolutely. And in fact, there's a new emerging science of how do you sample.
Right. I have 2 recommendations for listener picks of the week. 1, to learn about how a Californian coup d'etat established their wines as equals to French winemakers. Watch the movie Bottle Shock, which combines fact and Hollywood sensuality. Watch the trailer here. To read more about the entire process of making wines from growing grapes, figuring out when to harvest them, and fermentation, I recommend the book From Vines to Wines, and provides a link for both. In closing, In Vino Veritas. Mark, P.S. Feel free to use the grape image. The catalog image is technically copyrighted, but though I doubt Gusmer would care if you wanted to post it. This picture of grapes is particularly cool. He sent a picture of a half a ton of grapes, beautiful. Purplish grapes with that whitish powder on them, and we'll post that in the show notes. So thank you, Mark. I've learned a great deal, not only about endocarditis today, but also about winemaking, and I'm getting very thirsty. Anyone want to have a glass of wine after TWiM?
That would be nice.
Well, I'm here all alone.
Could we listen to a little Frank Sinatra?
Yeah, we could.
Absolutely. I have it on my iTunes list. No problem. Great idea. That is TWiM 63. You'll be able to find this at iTunes and also at microbeworld.org/TWiM. And if you like what we do, one way you can help us is to go on over to iTunes and rate the show. It helps to keep us visible in the crowded iTunes directory, which recently Apple announced that there have been 1 billion subscriptions to podcasts in iTunes. So there's a lot of competition there, so you have to help us out. We like to get your questions and comments. Send them to twim@twiv.tv. Michele Swanson is at the University of Michigan. Thank you, Michele, for joining us today.
Thanks for having me.
Michael Schmidt is at the Medical University of South Carolina. Thank you, Michael.
Thank you, Vincent.
And I am Vincent Racaniello. You can find me at virology.ws. Just want to let everyone know that a big ASM meeting is coming up in September. It's called ICAC, and I'm not going to tell you what it means because I always mess it up. Interscience Conference on Antimicrobial Agents and Chemotherapy. There you go.
You got it right.
I did. I screwed it up on a live podcast once. It was very embarrassing.
Oh, my.
Anyway, we'll be doing a TWiM and a TWiV. Michael will be joining me for a TWiM, and I'll be doing a TWiV. And Michael will also be doing ASM Live, Michael Schmidt.
Yes.
That ought to be fun.
I am looking forward to interviewing my colleagues and learning what's cool about their science, and it should be loads of fun.
And I'll be in the audience heckling you.
I'm certain of that. Between you, Ray, and Chris, I'll be on my best behavior, and we're going to be administering oxygen to Jim, and the press agent for ASM will be administering oxygen to him.
So if you are at ICAC, you can go to these presentations, but if you're not there, they'll all be live streamed. It'll be free. You can find out the times and the dates at microbeworld.org/ASM Live. And I'd like to thank the American Society for Microbiology for their support of TWiM, and in particular Chris Condayan and Ray Ortega for their technical help. The music you hear on TWiM is composed and performed by Ronald Jenkins. You can find his work at ronaldjenkins.com. Thanks for listening, everyone. We'll see you next time on This Week in Microbiology.